Hello, everyone. Welcome to the Genetics and Genomic Virtual event. My name is Yanfang Jiang. I'm a senior scientist with MilliporeSigma based in Missouri, St. Louis, Missouri. Together with my colleague, Michelle Ding, a scientist in our group, we will be talking to you about MilliporeSigma's newest strategies on a CRISPR-Cas9 toolbox with utilizing visualization and increased chromosome accessibility. Please feel free to submit questions during our presentation, and we'll be following up with you through emails afterwards. During the first part of our talk, I will first to give you an overview of our company's CRISPR portfolio. Then I'll introduce to you our newest Cas9 GLP fusion product, which can be used for visualization of the CRISPR system. Sigma-Aldrich is a pioneer as a commercial provider in targeted genome editing tools, with over 14 years experience starting from the first generation genome editing tools such as CRISPR-chrom and zinc finger nucleus. Sigma-Aldrich has become a prominent leader in the field of CRISPR-Cas9 system development. We have our first CRISPR-Cas9 product launched in 2013, and since then we have continued to offer to our customers a great variety of CRISPR products, including some of the award-winning technologies such as the CRISPR epigenetic activator in 2015, the renowned Sanger CRISPR libraries in 2016, the CRISPR-SAM libraries in 2017, and most recently, the proxy-CRISPR in 2018. The Sigma-Aldrich CRISPR portfolio consists of a variety of delivery formats. Lentivirus carrying the CRISPR systems can be transfected into almost all cell types, especially those hard to transfect cell lines. Lentivirus is also the carrier for our renowned Sanger CRISPR libraries. We also offer a great selection of plasmid carrying the CRISPR systems available for purchase at both the pre-designed form and the custom-designed form. The plasmid DNA can then be transfected into the desired cell lines. The CRISPR system encoded into the RNAs are also available for embryonic microinjection. Purified CRISPR-Cas9 proteins, together with the guide RNA, can form a protein RNA complex, which we call RNP. So the RNP delivery format has been more popular and preferred in recent years. And the RNP can be either microinjected or transfected into the desired cell lines. And this is going to be the major focus for my talk today. On top of the individual delivery format, Sigma-Aldrich also is the product provider for the whole genome CRISPR libraries, either in the arrayed form or the pooled form, including our award-winning technologies such as the Sanger genome, whole genome CRISPR library or the CRISPR-SAM library. SB-Cas9 is the first discovered CRISPR system and has been widely used in the genome editing field. Also, the RNA protein RNP complex is the preferred method for delivery in the recent years. Our Sigma-Aldrich SB-Cas9 is purified from E. coli and has a nucleus localization sequence fused to C-terminus to ensure efficient nuclear delivery into the cells. The purified SB-Cas9 together with guide RNA can form the RNP complex to be transfected into the cell lines. The guide RNA can either be in vitro synthesized into a single strand, or it can be ordered through our MilliporeSigma sigRNA system. So the sigRNA can either be one single strand sigRNA, or it can have two parts, the universal tracer RNA together with the CRISPR RNA that's complementary to the target strand. cGRNA can be easily used to form RNA complex with the purified CRISPR-Cas9 and ready for transfection. Using the RNP delivery format has the advantage of possibly eliminate the unwanted random integration on the genome, therefore reduce off-target effects. Our cGRNA system is convenient, efficient, and hassle-free. On the customer side, there's no cloning or in vitro RNA synthesis is needed. And also we provide a pretty short lead time, about 3 to 5 days, you will be getting your RNA. And also the RNA can be chemically modified and scaled up. Sigma Aldrich has a comprehensive Cas9 protein portfolio. The canonical wild-type CRISPR-Cas9 can be used for standard genome editing and cloning applications. We also have 2 versions of improved CRISPR-Cas9, the eCas9 and paired NICAS, both of which have higher fidelities that can be used to reduce off-target effects. We also have a special FN-Cas9 that we cloned and purified from a bacterium called Francisella novacida. And the FN-Cas9 can work as a programmable restrict enzyme for cloning large genome fragments into the vectors. We also have the catalytical inactive dCas9 fused with flag tag and belting tag that can be used for targeted genome binding isolation and enrichment. The dCas9 is also the essential component for our proxy-CRISPR technology, which is a novel technology developed by MilliporeSigma for increasing the editing efficiency for some non-canonical CRISPR systems. Lastly, we're going to talk to you about our new Cas9 protein product, which is the Cas9 GAP fusion protein that can be used to visualize the CRISPR system inside the cell. Before we designed our Cas9 GAP fusion proteins, we first took a look at the available similar fusion protein products available on the market from other suppliers. And it turned out that all of those fusion proteins failed to give good editing activity inside the cells. So CRISPR-Cas9 is a rather large protein at 163 kilodalton. Fusing a 27 kD GFP may most possibly compromise the folding of both the Cas9 and the GFP. Therefore, you have decreased nucleus activity and decreased fluorescence. And we suspect that this is probably the reason why the similar fusion products from other suppliers fail to have good activity. To solve this problem, we decided to design a specific linker sequence between the GFP and Cas9. The idea is to put the 2 protein parts at optimal geometrical distance so that each of them can fold properly and giving good activities. We had to actually experiment several different linker sequences and with their relative positions to the Cas9 protein. We also engineered 3 optimized nuclear localization sequences for enhancing the nuclear delivery inside the cells. Finally, we also carefully designed the expression and purification system so that we can make sure that we have high yield and high purity in the protein production. In this slide, you can see some beautiful green colors here. The strong green fluorescence was visible throughout the expression and purification procedure. And in fact, the green cell palette became a good indicator for us as to whether the expression level was good or not. The middle picture shows you the binding of Cas9 GFP fusion proteins to the nickel NTA region. So the nickel column, the natural color is light blue. Upon binding the Cas9 GFP fusion proteins, the region becomes strong green color. And when the fusion protein is eluded out, the resin turns back to the light blue. On the right, this figure shows you 4 individually purified fusion proteins that we have, each with a different linker configuration. You can see all of them at 5 milligram per ml. The differences in the fluorescence intensity indicate that indeed the linker sequence has effect on the protein folding. And we decided to choose the one with the strongest fluorescence for our production. We also want to make sure that our fusion proteins have good editing activity inside the cells. Since Cas9 cut different chromosome targets at different efficiencies, and the efficiency can also be different in different cell lines, we tested 10 target sites in multiple cell lines and examined both the on-target and off-target activities. Because we want to make sure that fusing the GFP to the wild-type Cas9 didn't alter the off-target property of the enzyme so that there's no more off-target effect generated. We assayed our editing results by both T7 even gel assay, which is a semi-quantitative assay, and also with next-generation sequencing, which is a more quantitative way to assay our editing efficiency. This slide shows you this is just a subset of the editing efficiency data that we have. This specific experiment was done in U2OS cells by RNP transfection. And what you can see here, the left 2 set of data are the 2 Cas9 GFP fusion proteins from 2 other providers. They both have really low activity across all 4 targets. The middle 2 sets are comparing our Cas9 GFP fusion to the non-fused Cas9, wild-type Cas9 counterparts. And you can see the Cas9 GFP fusion proteins has the same level of activity comparing to the wild-type on all 4 targets. The right 2 data sets are comparing the eCas9 GFP fusion to its eCas9 non-fused counterpart. So the reason we also wanted to make a eCas9 GFP fusion is that for some customers and some specific applications, they prefer to use the high-fidelity eCas9 to reduce the off-target effect. I want to point out here that the on-target activity of eCas9 is in general lower than the wild type, especially in the RNP format. And you can see from our data that our eCas9 GFP fusion is not only having a similar activity to its non-fused eCas9 counterpart, it also has higher activity than the 2 wild-type Cas9 GFP fusions from our competitors. So this set of data clearly demonstrates that our Cas9 GFP fusion proteins have superior activity, outperforming the other similar fusion products available on the market. One application for the Cas9 GFP fusion is to visualize the fluorescence inside the cells. In this set of microscope pictures, what we can see is that right after the transfection, the fluorescence is visible inside the cells, but the signal is mostly in the peripheral side of the cells. Then when time passes, the fluorescence becomes more centered in the cells. To summarize, Sigma-Aldrich is a brand leader in genome editing technology, providing a comprehensive portfolio of CRISPR-Cas9 products. Our new Cas9 GFP fusion proteins offer superior editing activity, outperforming similar products from other providers. The Cas9 GFP fusion proteins are great tools for visualizing the CRISPR system inside the cells. And now I'm going to turn it over to my colleague, Michelle, who is going to tell you our novel CRISPR technologies developed in MilliporeSigma.
Hi, everyone. My name is Michelle Ding, and I'm a research scientist at MilliporeSigma. And today I'm going to talk to you about some novel CRISPR technologies developed by scientists at MilliporeSigma. And as Yanfang has just talked about, the CRISPR system has become the rock star of the science community in the past few years. And it has a wide range of applications from food industry to agriculture or to pharmaceutical and medical field. And although this technology is very advanced, but there are still some hurdles that preventing this from unleashing its fullest potential for us to use. And these hurdles include some off-target cleavage or some low editing activity in those difficult to cut genomic regions or in those hard to transfect cell lines. So currently, there are several ways for us to improve the CRISPR editing activities, and this includes the introduction of the nuclear localization signal, or using a chemically modified single-part guide RNA to enhance its stability, or we can adjust the RNA and protein ratio in RNP transfection to maximize the editing efficiency. And today in my talk, I'm going to explore this field from a different perspective, and especially on how can we improve the editing efficiency on those difficult to cut genomic sites. And the two technology I'm going to covering today is the proxy-CRISPR and the CRISPR-chrom, and both of them have been published in the peer-reviewed journal, the Nature Communications and the CRISPR journal. So before we begin, let's talk about some basic science. So we all know that each human cell has 23 pairs of chromosomes for a total of 46. And some cancer cells has 3 sets or even more chromosomes. And actually, if we line up all the DNA molecules within a single cell, and the total length can be over 6 feet. And it's very fascinating to imagine how can a single cell pack such enormous genetic information in a tiny nucleus, which is often 5 micrometers wide. And actually, the secret to this super packing system is the chromatin structure. So if you look at this illustration, the DNA first wraps around the histone protein to form the beads on a string, and then it forms the helix fiber, and then the supercoil, and eventually it forms the chromosome structure. And if the DNA is not being actually transcribed, it remained tightly packed in this system. So, for the cell itself, this super packing system creates a lot of advantage for them and saves a lot of space. But for us scientists who wants to manipulate the DNA, it creates a problem for us. Because if CRISPR doesn't even have access to the DNA, of course, it cannot do the job. And in this slide, I want to give you some perspective on how the genomic sequence or structure can affect the Cas9 cutting capabilities. And in this experiment, we designed dozens of guides on 2 genes, the CAR and the POR genes. And then we compared the cutting capability of both FN and SP Cas9. So Yanfang has just touched upon FN Cas9 a little bit, and I want you to know that, so previously, people only observed the cutting ability of FN Cas9 in test tubes, and it remain widely inactive on most of the target sites. And that's kind of consistent with our observation here. So among most of the guides that we tested, the cutting property of FN is very distinctive from SP, and it remained kind of inactive on most of the sites, but on some sites it showed some activity. And another striking observation I want to make out of this figure is that if you look at some of the guides that within only 50 base pair range, the activity varied a lot. And it can vary from almost no cutting to 40% cutting. And these observations really led us to think, how can we better overcome the inhibition from the genomic structure of the DNA to better facilitate the Cas9 cleavage. And in this proxy-CRISPR technology, we leveraged the help from the two dCas9 proteins. And the dCas9 by its name is dead Cas9. So the dCas9 still has the ability to bind and unwind DNA, but it does not have the cutting function. So if we leverage the help of the two dCas9 to open up the DNA on the two ends of the interest of the sequence that we want to edit, it will make way for the major Cas9 to come in and do the real job. And so in this experiment, we also chose the FN Cas9. And as you can see in the figure, that on the second column, if by the FN Cas9 itself, it doesn't cut at all. And when we have either of the dCas9, it doesn't cut either. But when we began to combine the dCas9 to the FN Cas9, we began to see some cutting activities. And when we have two of the dCas9 present, the cutting activity has been significantly improved. And another thing is to achieve the best results for the proxy-CRISPR technology, usually we recommend designing your guide sequence for the dCas9 about 20 to 30 base pair from the PAM sequence. And in the CRISPR-chrom technology, we want to explore whether we can leverage the function of the chromatin modulating peptides to enhance the Cas9 cutting activities. And the idea is that by having the chromatin modulating peptides, we hope they can interact with DNA and also make it more accessible to the Cas9 proteins. And we fused several chromatin modulating peptides to both the N-terminus and the C-terminus of the proteins. And by testing 33 constructs, we eventually narrowed down the candidate to 3 constructs, which are shown on the slides here. And for all these 3 constructs, they all have the HN1 motif on the N-terminus, which is the human high mobility group protein. And on the C-terminus, we have 3 different modulators. And it's HB1, that's the human high mobility group, box A domain, and H1G, the human histone H1 central globular domain, and the CHD1, it's the East chromatin remodeling complex DNA binding domain. And for the rest of my talk, I will be referring them to the CMP. So here we select 4 difficult to cut genomic sites and compared the CMP fusion constructs to the wild type. And the indels or insertion deletion percentage was detected by the mismatch assay. And as you can see in the figure that for the 4 low activity target, the wild type cuts, the activity cuts from 2.2% to 4%. So it's pretty low. But with the presence of the CMP fusion, we see that the cutting activity dramatically improved with 3 to 6-fold. And then next, we used deep sequencing to look, had a deeper look at the mutation profile generated by the different constructs. And we can see in the figure that the mutation profile, the peak sequence, the peak position along the edited region pretty much aligned with the wild type protein. So this indicates that there is no influence, or the CMP fusion doesn't cause any change to the wild type in the NHEJ repair pathway. And then in this experiment, we expand the guide to 12 sites. And we selected 6 targets from the EMX1 site and also 6 targets from the POR genes. And we also narrowed down the CMP fusion construct to just 1. And the reason is because that for the third construct, the HN1CHD1 is the largest among all. And Yanfang has briefly touched about the size issue of SP-Cas9. because CRISPR-Cas9 itself is already a very huge protein. And when we add more motif to it, the size might become an issue in some downstream applications. So the smaller, the better. So we eventually decided, to choose between each the HN1 or HB1 or the other HN1, H1G. And among these two constructs, the HN1, HB1 construct performed slightly better than the other one. So we chose that to proceed with this testing. And as we can see here, the target activity was selected, was expanded from low activity. And here it's some of the targets have low and some have high to moderate cutting activity. And we can also see that for the low activity target, the wild type, the presence of the CMP fusion dramatically increased the cutting percentage. And if you look at the PORSPY6 target, the wild type cuts at 7%, and the CMP fusion protein cuts at 38.8%. So it's five-fold increase. While if the target itself has high activity to begin with, such as the EMX1 SPY10 site, the wild type cuts at 41% editing, while and then the increase in the editing efficiency by the fusion complex is not very significant. And this might be because when the guide itself is already active, then the system might be saturated, and then it's just leave little room for the CMP fusion to do its job. And in this slide, we set out to compare the HDR and NHEJ repair pathways, and we were just want to explore whether this CMP fusion strategy can be used to facilitate the HDR-based precision genome editing. And both HDR and NHEJ events were evaluated through deep sequencing here. And we introduced a single stranded DNA donor with a Hindi-free digestion site for the downstream analysis. And as you can see here, so the percentage of HDR and NHEJ event have dramatically increased with the presence of the fusion proteins. And that's also because the overall improvement on the editing efficiency with the fusion strategy. And if we compare the HDR versus NHEJ ratio, so for the wild-type Cas9 and the HN1, HB1 protein, the two protein, the HDR and NHEJ ratio remains around 1. And for the HN1, H1G construct, the HDR versus NHEJ ratio is around 1.5. So it means that these constructs actually preferred HDR versus NHEJ. And then in terms of the HN1 CHD1 construct, the HDR ratio versus NHEJ actually dropped to 0.6. So it means this construct actually prefers NHEJ repair pathway. So all of these results imply that this CMP fusion strategy can potentially be used to improve the HDR-based precision genome editing. And after our initial success with the CRISPR-Cas9, we next want to explore whether the strategy can be applied to other novel Cas9 systems. And the reason why we want to use some novel Cas9 system is because they usually have a different PAM sequence. And although the CRISPR-Cas9 usually is the most popular protein on the market, its PAM sequence NGG sometimes can set a lot of limitation when it comes to the target sequence selection. So having other novel Cas9s that can recognize a different PAM sequence give us more bandwidth in target selection. And sometimes it can also help to reduce the off-target cleavage. So for the SPA-Cas9, it's a smaller ortholog than SP-Cas9. And I just talk about the size issue. So this protein, the smaller It's easier for us to express and purify it, and it also works better with the viral vector delivery. And people through previous studies have also found that the CRISPR-Cas9 has a 6-nucleotide PAM sequence, so it's NNGTGA. So for the 6-nucleotide sequence, it can really dramatically reduce the off-target cleavage. But the problem for the CRISPR-Cas9 is that on previous studies, people only observed cleavage activity in cell-free conditions. And so in human cell, it hasn't been seen to be working. So we're very curious to see whether the CMP fusion can be used to facilitate the editing activity of the CRISPR-Cas9. So we fused the CMP fusion to SPA, and we also made a little modification to the sgRNA scaffold to make it more stable. And among the 4 targets that we tested, we were really happy to see some cutting activity of the CRISPR-Cas9 at a moderate level. And with the presence of the CMP fusion, the cutting activity further improved by 2-fold. And we also examined the mutation profile of the CRISPR-Cas9 and didn't see any difference between the fusion constructs and the wild-type counterpart. And we also delved into the HDR and NHEJ events after the editing, and also see an improvement on the HDR ratio. And for the CRISPR-Cas9, however, the HDR versus NHEJ ratio remained around 1 between the wild type and the CMP fusion. So it means it didn't have any preference to choose either of the repair pathway. And I just talk about sometimes that the CRISPR-Cas9 can set the limitation for the guide selection. And here is an example. So sometimes we have to be very specific on what sequence we want to edit, especially in these disease mutations. And here in this skeletal malformation associated mutation, we want to edit the mutated sequence, but there's no NGG PAM for CRISPR-Cas9 to recognize. So now it becomes very imperative for us to have another set of Cas9 that can recognize different PAM sequence for us to edit. So actually in nature, there have been about 200 Cas9 proteins be identified, and each of them usually recognize a different PAM sequence. And the idea is a little bit like when we use restriction enzyme. So you have a library of different enzymes that can recognize different sequence. And so if we can have that in a CRISPR system, and it can greatly expand our capacity in designing the guide sequence and our ability to edit the DNA. And at MilliporeSigma, one of our colleagues worked with some novel Cas9s, and he identified the new PAM sequence in 4 new novel Cas proteins. And you can see here on the slides. And I want to point out here that for the PX-Cas9, it cuts at the non-target strand 6 nucleotide upstream of the PAM sequence. So when the PX do the cutting, it will create a overhanging sticky end rather than a blunt end generated by most of other Cas systems. And then we're curious to see whether the CMP fusion can be applied to these novel Cas9 system as well. And so we fused the motifs to the 4 novel Cas9s, and then we were also happy to see that it can facilitate the cutting activity as well. So to wrap up my talk, so the CRISPR-chrom strategy is effective in improving gene editing efficiency, and it has the potential to improve HDR-based genome editing, and it is also a powerful tool to expand our research capability on some of the novel Cas systems and other applications. And we also didn't see any significant increase in off-target editing by using this strategy. So MilliporeSigma has been one of the earliest innovator in the genome editing field, and we're continuing to play a significant role in this field, and we're developing some novel technology for the future as well. And that wraps up my talk, and with that, we'd like to take some questions.